I'm Stephen Larson. Very nice to meet you. I got my PhD in neuroscience about 13 years ago at UCSD. I was working on informatics projects, neuroinformatics, back in San Diego. And before that, I studied computer science and I did software engineering. So I kind of come from a world of combining those two skills together. After getting my PhD, I started a software company as a scientist who felt that there weren't good enough software tools that were out there for the doing of science. And we started the company. We didn't take any investors. We didn't do it because we thought we were going to make a lot of money, but we did it because we realized that there was like just not good enough ways to get software projects done in neuroscience and life sciences in general. So today we're going to talk about, how to make neuroscience research better through collaborative data science. And we're going to talk a little bit about that. Let me allow Phil, who's also going to come in here, to introduce himself as well. Let him do that right now.
Hello, everyone. Nice to meet you. I'm Phil Dong. I used to be a grad student with Denise Kai working on calcium imaging analysis, and now I'm a data scientist at Metacell, and I'm handling a lot of our requests on the cloud workspace, the remote data science front of things. And well, I guess you'll hear more from me later. I'll be talking about specific projects I've been working on under this umbrella of cloud workspace. Yeah.
And Paolo, since your face is on the screen, you might as well introduce yourself as well.
Yeah, sure. Good afternoon, everyone. Nice, meeting you virtually, I suppose. My name is Paolo. I'm based in London, UK. I'm part of the commercial team at Metacell, and I help organize the talk today. And thank you for coming, and hopefully more will join us later. This is off the back of a sponsorship that we did this summer to the Bomb Brain conference. I know Stephen had the chance to come and visit your campus, which is great. And I hope you have a good afternoon and a good chat with Stephen.
And if you find the food tasty, you can thank Paolo.
Yeah, and then next time I come and have some as well.
All right, cool. So with those intros, then let's kind of shape the talk. So I will talk very, very briefly about what we do in terms of building custom software for neuroscience. But then the main thing that I want to talk about is this remote data science opportunity that we've been offering. And then Phil will go in and explain that a bit more. And we will use examples from actual scientific work that he's done in the domains of calcium imaging, photometry, and extracting behavior, things that are kind of important these days for neuroscience experiments. So you're going to hear a bit about science. You're going to hear about some best practices, basically. And then, again, sorry for confusion. That the design is that I would speak for about an hour, and then the goal is to do a roundtable actually, and to go around and to talk to each of you about what data science challenges you might be facing today. We want to learn, and then if you're up for it, we might be able to help you. But that's so there's an interactive session intended for after this. Okay, so custom scientific software. So again, really quickly, we've been in business for about 13 years, and we've had the opportunity to work on really cool projects that are both brain related and not brain related. What you're seeing on the screen right now is a project we did with a big pharma company back in the States that allows for combining two-dimensional and three-dimensional information around the structure of the human brain. So here you sort of see both anatomical coordinates as well as some graph structures that are put together along with some activation areas. So custom software like this, we build, put it on the web. It's all web-based. We build it for the purposes of having data shared. And we've been doing this now. We're building a zebrafish atlas with another one of the Max Planck Institutes, actually, here in Germany. And so it's combining our 2-dimensional and 3-dimensional work. We also do work with the Allen Institute. We do work with Chan Zuckerberg Initiative, things you guys have probably heard of. We've worked with the University of Edinburgh, University College London. So yeah, a fair bit of projects like this. This is the virtual flybrain that we worked at with University of Edinburgh, which again also combines 2D and 3D data, microscopic data, also information about what types of things are called in terms of ontologies, references out to papers, scientific papers. So this fuses together a lot of data sets together and allows for folks to look up, you know, in a combined way what's going on with the anatomy of different animals, in this case, the Drosophila. And over time, as I mentioned, we've been in business for 13 years, so we've had a chance to work with lots and lots of organizations. Like I mentioned, big pharma companies at the top, lots of academic institutions, some nonprofits in there, a few medical hospitals, this one here is in Toronto. But yeah, we've been able to do projects like the ones that I'm mentioning for a bunch of different organizations over the years. Okay, and a lot of times people ask, you know, so what kind of data do you deal with? Generally, the answer is like any of it. You know, our observation is that life sciences and neuroscience in general has been moving so quickly that there's always a new data format, there's always a new device, there's always, you know, microscopy moves forward in different ways. And so largely, you know, these are some of the examples of data that we've worked with, but we basically, because we understand the basics of computer science, we will work with any data format that you can encode in, you know, a binary format, and work for better ways to share it, create data pipelines, create visualizations. Okay, so that's the custom software. Now let's talk a little bit about data science support. So we found that there has been a pain point that researchers have, which is that, you know, you've done your experiments, you've, I don't know, whatever animal you might be investigating, be it a mouse, be it a fish, be it a C. elegans. You do some work, a lot of work to extract data, and then you reach a point where it's time to go run some statistics, do this sort of thing. Or maybe there's a new statistical method that's out there, a great paper just got written about it, you'd like to use it, or you read about some Python library that just got published that you'd like to work with, or something like this. So you have some particular analysis completed, you've got data, but you don't have expertise in the lab necessarily to take advantage of that next new analysis technique, or ask that statistical question that you want to ask. So there are a few options. Maybe option one is you bring in a student or a postdoc, you get a collaborator with a computer science department, or you bring in a, like I said, a postdoc if you're a PI, or you work with a master's student, somebody else that, you know, has more maybe computer experience. But that can be time consuming. If that's a thing where you have to hire somebody, there's a hiring process involved, and that person may not have a specific focus on the thing that you want to get done. So another option that we've been finding folks like is to find a data scientist who has that expertise, but may just not be physically near you. Someone who's like really good at the thing that you want to do, but they might be in some other part of the world. And they, you know, maybe can come in and just do the work that you need for a limited time and then kind of go away. So they don't have to like join your lab, or you don't have to hire. There doesn't have to be a big process involved. They come in, they help you, and move on. So that's a bit what we've been working to help folks with. So we've been experimenting for a bunch of years on what helps scientists solve this problem the best. And so we've figured out that when we pick a computer that sort of lives remotely in the cloud, which we call a cloud workspace, that can provide a platform that helps to deliver remote data science to scientists. The idea being that that computer up in the cloud becomes this place that both a data scientist and experimentalist can log into, and all the code that gets written still stored in something like a GitHub, but it all kind of lives there in that thing. the data can also be interfaced up there. So analysis happens in that space. It's a collaboration space. It solves the problem of like all the analysis is on your laptop, you know, and then somebody has to log into it, or it's on somebody else's computer and you have to log into that. It is that this is a space that both scientists and data scientists can connect to. And so in our version of it, there's a virtual desktop that you can log into. So you can access it on your laptop, but you're like logging into this remote machine quite easily. And we use also Jupyter Lab to help build those analysis pipelines, which is a popular open source software in Python. And that allows for specific data science problems to be solved as needed. And allows for multi-user collaboration, as I mentioned. So what does it look like? So I think we've been on all of us have been on a lot of Zoom calls, I imagine, in the last years, right? It's a pretty common thing. So it's basically a Zoom call. There's Phil. We'll talk to you later, but it's basically collaborating around, you know, your data and writing scripts either collaboratively or looking at the results of them together, and talking about them and getting on a call once a week or once every 2 weeks. So in the case where the data are relatively small to medium, things that you can collect easily in the sort of gigabytes up to sort of low terabytes, right? It's easy enough to just send that data from, say, a laptop computer or maybe the data collection computer that's sitting in the lab. We also are experimenting with ways to deal with even much larger data sizes. So in some cases where there's terabytes and terabytes of video data, there are ways that we can set up a remote connection into, say, a network addressable file system and still be able to access it while leaving most of it in place. So just addressing that particular concern. Okay, and so what we found is that there's sort of 4 ways to use cloud workspaces to support research, but I'm really just going to focus first, which is really remote data science. The others, remote data standardization and sharing, is kind of what I mentioned, that like more and more there is emphasis on standardizing data and sharing it. I don't know what the rules are here in Germany. Back in the States, the NIH is getting more and more insistent that data sharing actually happen, not just claim to happen. So we work with some scientists to help package up data in standardized ways, submit to standardized repositories, that sort of thing. That's also a thing that we can do. Also, we find that in educational workshops, the workspace is very useful because, like, if everybody needs to learn the same software and like you all had your laptops here, you all could log in to a workspace and all actually be using the software immediately without having to install it on your local laptop. Saves a lot of time. And then finally, with collaboration. So if you had some key data set and you wanted other collaborators elsewhere in Europe or back in the States or wherever to see the same data, they also could log into this workspace. So just a few different ideas as we go into 2025 and beyond of ways that we can be using cloud workspaces to do more scientific collaboration. It's certainly easier than like mailing a hard drive across the world, which I know many PIs still do. Okay, so now, so now let's get into some examples. And for this, I'm going to go ahead and bring Phil over into view. There he is. All right, I'm gonna full screen this. I'm gonna try to turn around the camera so Phil can actually see you a little better. All right, cool. And then screen share, I will sit down.
Does it work? I couldn't really tell. I hope. Perfect. All right. Okay. So jumping into this remote data science part of the work. Basically, there are 2 modes of operation. for remote data science. The first one is custom analysis performed on behalf of scientists. So the scientists ask a question, and we help answering the question by performing analysis on their data, either on the neural data or on the behavior data. The other mode of operation is we build pipelines so that the scientists can run by themselves. And then we can customize the pipelines to suit the specific needs of specific projects. As Stephen mentioned, currently we focus on 3 area of data of neuroscience regime. Focus on calcium imaging, photometry analysis, and then behavior extraction. And we'll go into a little bit detail into each of this later. So here, oops, okay, gonna show all of them. So here are some example projects we have worked on before in the cloud workspace. And this kind of show you the breadth of regime we work with, and also the breadth of institute we collaborate with. So for DGIST in Korea, we work on investigating neural activity during a reversal learning task, and specifically we look at reward signals and how they interact with animals' choice during the learning of the reversal task. We collaborate with McGill and work on investigating inhibitory control in go and no-go task, and specifically we look at how the inhibitory signals correlate with animals' behavior in the no-go trials of the task, and we see how they change across different phases of learning. And then we work a lot with a Max Planck Institute back in the States, the Max Planck Florida Institute. With one group, we investigate a cell type specific response during social behavior. We'll jump in more detail right after this slide. With another group, also at MPFI, we investigate the internal state change of animal during a feeding behavior, specifically how the population activity change when the animal is more hungry compared to when the animal is more thirsty, basically. So that's kind of the, I think it's pretty common, and it's kind of the range of neuroscience questions and project we work on. So here is a little more deep dive into one of the projects I just mentioned, MPFI. Here, the Dr. Ryohei Yasuda group at MPFI developed a technology that can record calcium activity when the animals are freely behaving in a social environment. And then they can image the same field of view under a tabletop confocal microscope so that they can identify different cell types and projection information by combining the virus labeling here and the multi-channel imaging in the confocal. So that's the basic idea. The scientists developed this technology. They performed the experiment. But then after that, they need some expertise to deal with this very complicated data set, right? You have the activity of neurons, and you have their cell type information. How do you, and you have behavior, right? How do you deal with them? So this is where we jump in to help. And our task in this project is to find distinct response to distinct behaviors for distinct neural populations. So that's our basic task that we're given. And as an example, here are some analyses we performed during this collaboration. We helped cleaning up the behavior data a little bit by normalizing and thresholding different metrics coming from different source, different behavior segmentation softwares. We performed the deconvolution on the calcium imaging data so that the estimation of neural activity are more temporally accurate instead of having a long decay tail. We tried some cell clustering based on their response to different events. So each point here is a single cell, and we try to see whether there are any cluster, there are any pattern, whether the same kind of type of cell have the similar response. And then we also perform some analysis where we try to cluster the whole population activity at every single time point. So here, each point is a represent the high-dimensional population activity at a single time point. And we color them by time, color them by behavior to see how they progress and whether they sort of change during this social interaction task. And here is the sort of the output of this collaboration. This is the figure that made it into the paper / preprint, and this is actually the last figure in the paper. So here in A we have some example traces from different cell populations, and they are differentially activated by social behaviors. For example, they are differentially activated by either aggressive versus non-aggressive interaction, or they are activated by familiar versus novel social interaction. So in A, we just have some example traces of cells. And then in B is where we have a breakdown and like an overview of response grouped by different cell population. So in the inner circle is the proportion of cells that are basically kind of projecting to different brain regions. And then in the outer circle, the number indicate how many types of behavior events they are responding to, right? So using this pie chart, you can basically have an overall understanding of how different population gets breakdown and how reactive they are to the social behaviors. In panel C here, we have basically a more detailed breakdown of the same thing. We have a breakdown of the relationship between the cell population and behaviors. In the top row here, we have different population of cells with that's colored by this different color here. And then at the bottom row, we have the different types of behaviors. And this line connecting them, the thickness of these lines indicate how many cells a particular cell coming from the same projection are reactive to this particular type of behavior. So this final figure here really highlight the strength of the technology, where it's clear you can use this novel technology to image and investigate new activities differentially for different cell population of different projection in the brain. So that's one of the highlight we have, in the sense that we basically perform the analysis for the scientist. And now we move on into the pipeline we built so that the scientists can sort of run on their own. Again,
you guys want to ask any questions, by the way, about anything so far?
Sure.
Like, we can go in here for a bit.
Yeah, but my questions through for the part before, so I would just ask them in the end.
Okay. Cool. Anybody else? Good. All right. Yep. Go for it, Paul.
Okay, cool. All right, so now we move on into this pipeline. And again, we focus on three main areas currently: calcium imaging, photometry, and behavior analysis. And basically the pipeline is based on open source packages, or they are done in collaboration with other sources. So the calcium imaging was based on the Minion package, that's open sourced. The photometry analysis pipeline is built together with the company that manufacture the hardware device, Neurophotometrics. And then the behavior analysis is built based on Simba package, which is also sort of use deep lab cut in the backend. And again, that's the three area we focus on. But when we go beyond this open source package, is that we can perform customization for the customers so that they suit their experimental needs better. Here is a quick look at the Minion pipeline. The pipeline was built using the Jupyter Notebook interface, where the user can see all the documentations, instructions, along with the code here, and they can run them in the same interface. And they can, of course, change the parameter, change the code, and then they can see the interactive visualization or the result of the code in the same place. So this whole thing makes the pipeline more interactive and user friendly. again, so it's still underlying it is still a bunch of code, but we found that it's easier for the user to run pipeline like this on their own. Here is an example customization we built in one of our collaboration. Here, our customer is actually an industry pharma company, and what they're doing is they are performing high throughput calcium imaging with different drug conditions on basically a lot of wells, right? They probably perform this on, let's say, 96 well plates altogether. So what they need is they really need an overview of the extracted neuron footprints across all the wells at the same time, so that they can quickly verify the quality of the analysis. So that during our collaboration, we build this interactive visualization here, where after running the analysis pipeline, they automatically get this view across all the wells and see whether all the cells are identified properly. And similarly, we've done this for the temporal activity for the cells, so that after running, they have this visualization of extracted temporal activity, and they can quickly get a sense of whether there are any interesting features in the cell activity across all the wells, and which you can sort of clearly see in this snapshot right here. There are some very interesting pattern. The cells tend to fire in some very regular intervals. That's sort of related to the drug condition they're testing. So that's an example of the calcium imaging pipeline customization. Here's another example of the photometry pipeline. So again, the pipeline is built on the Jupyter Notebook interface. All these plots are interactive, and right after the analysis, you can get a plot showing you the activity relative to different behavior events in the data. For the customization part, in this example, we collaborate with UAB, and we build a pipeline that supports FET3 device, which is an open source device for studying feeding behavior. So, we support for this type of timestamp so that you can look at the new activity when the animal are, having different feeding behavior, when they're eating versus maybe satiated, stuff like that. And lastly, here's a snapshot of the behavior extraction pipeline based on Simba. Again, we transform everything into a notebook so that after running it, you have this video here in the same Jupyter Notebook window. You can verify the result of behavior classification as well as the animal tracking, the point tracking. So that's sort of summarize the example of project we have done under the Cloud Workspace service. And in summary, we believe this service can facilitate and help scientists with their questions, either by running analysis on their data or by building the pipelines where they can easily run on their own. And with that, I think I'll hand over to Stephen for some closing remarks.
Yeah. Just wrap up a minute, and then we can do questions. So, thank you, Phil.
Thank you.
I'm gonna bring you back over here and switch back to this. Okay. And those are the same slides. Through, and then we get to this. Okay. So, yeah. So, in summary, we start off explaining to you a little bit about the company, where we got our start. Basically, we are scientists. We work in the open source. We want to help scientists. We talked about some of the custom software that we built at the very beginning that involves 3D brain atlases and such. And then as we progressed in the last few years, we wanted to get even closer to helping folks with doing the analysis itself. And so then that's what has led us to where what you see that Phil's been able to do. We're helping labs around the world get data science help they need. And so I'm here today away from Boston, Massachusetts to talk to you guys and see what that looks like over here and understand what your needs are. So yeah, and we've done work, as Phil mentioned, in calcium imaging and photometry and behavioral analysis. So yeah, okay. And you're in the conference room now, so perfect. Okay, great. So thank you guys very much.